Hello, my name is Karolis Ramanauskas. I would like to thank you all for choosing to watch my talk today, and I would like to thank the organizers for their hard work under these difficult circumstances. All right, let's get to it. Problems in ecology and evolutionary biology often necessitate sequence data. We use it to study genetic diversity, population genetics, species delimitation, and identification. This data is also critical in macroevolution, macroecology, community phylogenetics, as well as quantitative genetics and development. Until recently, there were two primary sources of sequence data: in-house sampling and sequencing, and low-throughput sequences deposited in public databases like GenBank, often as a result of studies aiming to understand the evolutionary relationships between organisms or genes. But there has been a vast transition, a technologically driven shift in kind and amount of data. Single gene submissions to GenBank have slowed and were replaced by large datasets produced by high-throughput sequencing methods. In just 12 years, the size of the Sequence Read Archive has snowballed. It now contains over 14 quadrillion open access bases. Roughly one-third of the SRA database is comprised of RNA-seq data, representing a wide range of taxa and almost invariably including many genes found in megaphylogenies. Although this data is free and available publicly, it is not necessarily easily accessible. For example, on NCBI's website, only a subset of all SRA datasets can be queried with sequence search tool BLAST. Additionally, assemblies are not available for many, if not most, SRA datasets. I took this screenshot a few days ago. It invites us to browse over 3 million pages of raw data. Because of these and other hurdles, the weight of the arcane set of tasks required to process raw read data often falls on the researcher. The purpose of Kakapo is to ease this burden. Kakapo is a Python-based pipeline for E&D workers who wish to squeeze more data out of GenBank. It relies on many tested and established tools and focuses on repeatability and ease of use. In the rest of my talk, I will first review the key steps of the pipeline. Then I will show you a demonstration of Kakapo. I will do a brief review of the input files. I will then show you how to run Kakapo using those files. And finally, we will take a look at the generated output. Kakapo simplifies finding one or more transcripts of interest in an unprocessed set of RNA-seq reads. However, if you have an assembled set of transcripts, you can use those as well. The pipeline requires two types of input: one or more RNA-seq datasets, and the description of one or more protein sequences you are interested in. This is done by modifying two template configuration files provided with Kakapo. Kakapo will download the raw read data for the provided SRA accessions, or it can use your own FASTQ files. The reads will then be error-corrected and trimmed. Optionally, the pipeline will map mitochondrial and chloroplast reads to the best available reference genome and create separate datasets for these organelles. Additionally, the user can provide any number of arbitrary genomes for mapping. This may be useful, for example, if you are interested in separating viral or bacterial reads. As a final filtering step, Kakapo will run Kraken2 to filter any other non-target reads. Proteins of interest, or queries, can be provided as NCBI accessions, amino acid FASTA files, PFAM protein family accessions, Go terms, or any combination of these. In the end, Kakapo will output several different files: FASTA files with assembled transcript, FASTA files with open reading frames, both as nucleotide sequences and their translations. Lastly, it will optionally run InterProScan and create complementary GFF files with predicted protein domain annotations. This was a simplified overview of Kakapo. A more detailed flowchart can be found in the file section of this talk, and it is also available on GitHub. What follows is a greatly simplified version of the analysis I performed on over 200 RNA-seq samples as a part of my thesis work in search of sRNase-based self-incompatibility system across eudicots. For background and a review of results in the family Cactaceae, please see the recording of the talk by my advisor, Boris Igic. Now let's take a look at Kakapo in action. First, I will briefly show you the structure of the two configuration files. I will focus on the input data, glossing over other parameters. Do not be alarmed, most of them are the default settings that come with the template files and hopefully are self-explanatory. The target SRA accession section of the first file allows you to list the RNA-seq SRA run accessions, one per line. In this example, I'm using a single accession that refers to RNA-seq dataset from Hylocereus polyrhizis, a species in the family Cactaceae. In the Target FASTQ files section, you can list your own RNA-seq FASTQ files. In this example, I am using 6 samples from 3 Schlumberger truncata individuals used for my thesis project that Boris talked about on Wednesday. These are paired end reads. Notice a star used instead of read 1 and read 2. Kakapo will automatically use the correct files. In this case, FASTQ files are compressed, but you can use uncompressed files as well. The target assembly section allows you to specify previously assembled transcriptomes. For these files, Kakapo will skip the read processing and assembly steps and will engage only the final steps of the pipeline. Finally, there are 2 filtering sections, Botany2 and/or Kraken2. Botany2 is used to filter out but retain reads that map to 1 or more reference sequences. In this example, I am providing a FASTA file containing a reference genome of cactus virus X and directing Kakapo to filter plastid and mitochondrial genomes. Note that for these organelles, Kakapo will try to find and download the best available reference genome or genomes from the RefSeq database. It will do this by walking up the taxonomy, starting with the species of the sample and expanding to the genus level and so on. Kraken2 is used in a similar manner and is helpful in filtering out ribosomal RNA or reads from non-target organisms. While I do not have the time to go into more detail, I just want to point out that each item in this list refers to a Kraken2 database. Some, like this mini Kraken2, which consists of various bacterial and viral sequences, is distributed on Kraken2 website, and others, like this human one, is custom built using the tools provided with Kraken2. The second configuration file allows you to define search strategies, which are used by Kakapo to search for the transcripts you care about. The example file here contains 2 search strategies, one for T2 type ribonucleases and the other for FBOX domain containing proteins. All these settings in combination define a single search strategy, and while I do not have the time to go into more detail at the moment, briefly, the purpose of each search strategy is to allow the user to tell Kakapo how to run BLAST and how the results should be filtered. To run Kakapo, you simply need to provide the paths of the configuration and the search strategies files. In this case, I am using an additional argument, force_deps, which tells Kakapo to ignore the system-installed versions of the various pieces of software the pipeline requires, and use the versions installed by Kakapo. I'm running on a compute cluster at my school, which has different versions of certain software, and this option makes sure I get consistent results regardless of what is installed system-wide. Before the analysis starts, Kakapo will check that all the required tools are available and will attempt to install the ones that are not found. At this point, I will spare you the verbose output and will fast forward to the end to take a look at the results. I simply dragged and dropped a few key result files from the Kakapo output directory into Genius. I did not edit them in any way. All the annotations were produced automatically either by Kakapo itself or by Kakapo running InterProScan. Recall that we used 2 search strategies, one for FBOXes and one for RNases. Kakapo produced 2 final files named accordingly. Let's take a look at the RNase results. I will turn on some of the annotations to help us make better sense of what we have. InterProScan predicts that we do, indeed, find T2 type RNases. Let's take a look at coding sequence prediction and switch on translations. I can quickly extract CDS annotations and align them using math. Let's build a fast tree. This is the self-incompatibility RNase clade. Notice 2 alleles per sample, indicating they are heterozygous at the S locus. Also notice the interspecific polymorphism and the large divergences between the alleles. The divergences of other ribonuclease paralogs are much smaller. Additionally, all three Schlumberger truncata samples share one sRNase allele, but not the second one. You may recall that I asked Kakapo to map reads to the cactus virus X genome, but I did not elaborate further. We noticed that some of our individuals were not looking so great, and perhaps unsurprisingly, we discovered many cactus virus X reads in those samples. Both styles and pollen of this individual, for example, contains millions of cactus virus X reads. Other individuals apparently are also infected, but the load is multiple orders of magnitude lower. Further, we looked at other Cactaceae RNA-seq runs on NCBI. The Hylocereus polyrhizis SRA dataset, for example, shows a high viral load as well. Curiously, the paper related to this dataset studied defense-related genes in response to the fungal infection by Neoscatalidium dimidiatum. The SRA run accession I analyzed in this example was labeled as normal or disease-free. However, I find that more than a quarter of these reads consist of cactus virus xRNA. Additionally, there appears to be very little plant RNA in it, although the authors labeled the sample as stem tissue. As my advisor says, every transcriptome is a metatranscriptome. The takeaway from this example is that we should practice caution when using SRA datasets. Labels may not represent the data. The last point I would like to make is that I designed Kakapo in order to quickly find specific transcripts given only raw RNA-seq data. In order to do this, I first wanted to have a pipeline to clean and standardize the reads. This process turned out to be quite productive in itself. We did not set out looking for viral infections, for example, but Kraken-2 identified a significant number of viral reads in some samples, and I subsequently added the Botany 2 step to map those reads. While Kakapo is in active development, I think it is quite powerful and customizable. I hope that some of you will find it useful in your own research. With that, I would like to thank Alexa Tishka, an undergraduate in our lab who has taken over the Cactus Virus X project, my advisor, Boris Igic, for support in and outside the lab, and finally, the NSF for funding my work.